Okay.
Yeah.
So since the beginning of the last year, we stopped most of the things that we are doing at the lab and start to perform the genomic surveillance in the state of Rio de Janeiro, where the state that I live, but also helping the other states in Brazil to doing the genomic surveillance for the SARS-CoV-2 genome. So the question that most of the people around the world are doing is to following the variants of concern or the variants of interest that the both can make. Huh?
Teresa, sorry if I stop you, but you should switch to the full screen visualization of the presentation, because we can see it, but not in as full screen.
No. And now?
Wait a moment, I guess. No.
Yes.
Now, yes. Okay. Thank you so much. And sorry for.
No, thank you.
Perfect.
But now it is much better. Thank you.
Okay, so we are working to in order to identify the variant of concern that can be associated for the behavior of the virus to increase the transmissibility, the virulence of the virus, and can be decrease the effectiveness of the vaccines and the test of diagnosis and the therapeutics. But also we are looking for the variants of interest that this is the main proposal. Two of these are the main proposal of the WHO organization in the world. So we start here in the Rio de Janeiro, the project that the name is Corona Única RJ. Sorry, some words are in Portuguese, but the most important that we aggregate many institutions in the state. We collect the sample. We have a group of the students and researchers that for many different institutions here in Brazil, and we start to sequencing the virus, but we are starting to do the metagenomic, the exome, and the transcriptomic analysis. As we have a supercomputer at the lab, so we use many tools with the big data or IA methods to order to analyze the data from those samples. So here is the state of the Rio de Janeiro. We are able since the beginning of the last year to sequence the genome from the whole state. Here is the capital, the municipality, Rio de Janeiro City. So we are sequencing according to the number of the population and the number of the case. Unfortunately, in Brazil, we had the sad number of 6,000 deaths the last week. So the number of the cases are increasing a lot in Brazil. We are still having a lot of cases of SARS-CoV here. So here is a map of what happened since the beginning of the pandemic period. We start from some variants, the number of decreasing, then we are another variant that has been discovered by our group. And now we can see here is the Delta is dominating the world. Here we can see better. Here is the P1 that has been discovered, the Gamma, that is identified in the north of the country in Manaus. But since the beginning of this year, it started to be the most frequent lineage here in Brazil. But now the Delta is increasing. We identify in the beginning of the June, and now is dominant in the state and start to be dominant all over the country. And to do the genomic surveillance, since the beginning, we can see that it's a dancing for the lineage occurring. This is a sample in my state, but if you can see changing by the colors, the number is changing, and some lineage is dominant for each other. It's depending of the period that we are analyzing. We are sequencing more or less 1,000 genomes per month, and we are able to identify many lineage or many sublineage of each one this lineage. So in in November of the last year, we identified the P2, that the name after the WHO are given the name the P2, but that was first identified here in Rio de Janeiro, and she spread, this lineage spread all over the country in 2 months. And we are also identifying the beginning of this year, the P1, that is the gamma variant in the state of Rio de Janeiro. For identifying this lineage, what we are doing many phylogenetic analysis. And as we can see here, an example that we have many of these in yellow is the P1, the gamma lineage, but we are able to identify sublineage. For example, this one in one city, in municipality here near the capital of Rio de Janeiro, that's name is Conceição de Macabu. When we look this cluster, we performing phylogenetic analysis, we can identify that there are some important regions, mainly in the spike protein, that is the region that's the protein of the virus that's important to connect to the, with the human cell. We identify that there are many mutations in these samples that arrive in this lineage. We submit that to the pangolin lineage, and this was described in new lineage. So we continue to do the sequence here, and we are able to identify many other lineage, such as this P5, that they have some mutations such as a A4084 key, and in the spike protein that also important to the immunological escape of the virus in relation from the human cell. And we perform to doing that, and we identify, as Brazil has a high number of contamination, we identify many different lineage that has been approved from the pangolin and from the other areas. To focus a little bit in the Rio de Janeiro state, we follow this lineage, the dancing of this lineage all over since the beginning from the pandemic, and we can identify here some holidays from as far from the election, Christmas, Happy New Year, Carnival, and we can see that the number of the case increasing after these periods of holidays. And we can see also that introduction of the new lineage that is predominant in correlated with each different holidays. And we can also see that identify that has been already described in the literature, that there is a portion that only 2% of the sample we are able to spread to carry a viral load high in the population. So it's very important to prove that is important. Oh, yes, and then we are able to identify many regions. And now here in my last slide, so we identify the delta in the state of the Rio. Is incredible that they start to identify the first in the June, and the end of August is predominant in all of the municipalities in Rio de Janeiro. So is that we are doing now. I cannot talk more about that. We are also doing the study, the microbiome in lung from patients from the care unit here in Brazil to identify many bacteria and virus and associated to the variants. It's that, sorry.
Tereza, thank you so much.